I want to thank the organizer to invite me to this very nice conference, because there is a lot of good information and very good science. It inspired me a lot because I'm going to present to you some very new data for a project, a very recent project. And I have a lot of difficulty to make sense about the data, to be honest. So I'm going to explain to you how could we test the impact of odors on social interaction in mice? And could the ultrasonic vocalization be helpful to try to get interpretation of that? So why odor? Is because you know that welfare is very important for animal and for lab animal as well as farm animals, all the captive animals. Because of course, during their lives, they're gonna have alteration in their possibly in their stress and in their anxiety during birth, during weaning, when you switch from mother to normal food without the mother interaction, and during some manipulation, of course. So we know that environment enrichment could be nice, but odor enrichment is not very well used, even if it has been proved to improve well-being, food intake, and cognitive performance. So we wonder if we could use some odor to increase welfare in animals in changing maybe their affective or emotional state, and by that changing how they behave socially, and to increase the welfare of the group, in fact. So because we know that some odor could convey very positive or negative value, like, you know, odor of predator, or for positive odor linked to maternal context. And for example, collaborators, Christine Bally, shows that if you present odor to a pregnant mother, the offspring, if you recall the odor to the offsprings, it would change their anxiety, so potentially triggering some emotional states. But before using this kind of huge emotional, possibly emotional odor, we want to know how positive odor may modify social interaction, and how valence of an odor may modify social interaction. If we guess that odor may change the emotion, for example, we have demonstrated in another study that if you stress an animal, a black cyst mice, and you do social interaction just after, it would increase, and it was expected, aggressiveness and dominance in their behavior. So maybe if odor is positive, maybe you would change the aggressiveness and dominance. So question in this very preliminary study was, okay, could we find some odor with different value? Could this odor influence social interaction? And we focus on adolescent animal. And could ultrasonic vocalization describing frequency frequency of vocalization could help us interpret the impact of odors? So for that, the first thing was to find some odorants who may have some value for animals. So we focus with the help of Christine Bally and her team to some data from Saravia et al. In PNAS. So they look for like 70 odorants, and they have done a battery of behavioral testing to try to guess that some odor may be attractive and some may be aversive. And they test a lot of odorants. For example, for aversive odorants, they test TMT, which is a classical predator odor, EBT, and others. And they look for investigation time. So as you could see here, for EBT, there is not so much investigation like predator odor, but if you just diluted EBT, it could become to be attractive. So it's also something hard to deal with because you need to be at a good concentration. So for our study, we try to choose, we choose, EBT at around this concentration. Ambretolide, which is a musk odor, have the advantage to be always researched by animal. So we use medium concentration. And for vanillin, vanillin could be neutral at this concentration. So from this paper, we decided to use three odorants, ambretolide at 39 micromolar, vanillin at 85 millimolar, and 2-isobutyltiazole at 70 millimolar. So we also choose this particular concentration for human to be able to scent this odorant, because if not, it would be very difficult to use it during behavior for the experimenter. And this one is smelling like, green leaf of tomato, and this is more musk and floral. And we changed the way of presenting odor to keep in other study I would not describe. And also to avoid animal taking a lot of time to search for the focal point of odor, we try to do some global odorization of the cage of animal. So we have done very simple test of preference before using the odor. On some animals, only 5 CD1 male and 5 CD1 female, in a very simple, choice place, in fact. So one place with odor, one place without the odor, and we look if there is a preference in calculating the ratio between time spent in this place compared to the sum of this place and the other one. And there is a lot of variability. If for female, you see here, EBT seems to be not really attractive, and vanillin and ambretolide neutral. For male, it was not that clear. It was maybe ambretolide more attractive, and the other not that, just neutral. So it seems, of course, we have only 5 animals, so we keep with this odorant to test. But we were, of course, wanting to use USV frequency to test if we could use it as marker of emotion, even if from all the other great talk, probably it's not that easy to use like that. And we would add a preference test for the odor at the end of our social interaction to try to see if these animals are preferring or not the odor. So how do we test social interaction? So from many times in our team, in the team of Sylvie Grano, we use this social interaction task who promote dominance. So you have the host mouse, which is put in another cage for 30-minute habituation. And after that, you put a visitor animal or guest animal during 8-minute social interaction. This animal is same sex, same age. And of course, we record USV. So I could show you how it looked like on the video. So here you see the top of the cage because we want to restrain the odor in this cage. And we have put different okay, it's like a little bit, but don't problem. Okay, so I hope you would, of course, it's not lagging like that during the real behavior. So we put the visitor one, we put back the top, and we score the behavior between this animal. And the interest of this task is that there is not so much aggressivity, even if in CD1 you could have aggressiveness, but in our condition, not that much. And also there are adolescent animals, probably that's why. So what do we measure in behavior? So we measure using a neurological keyboard, contact initiated by host animal, contact initiated by visitor one. Of course, in our condition, mostly the hosts are initiating the contact. And we also measure follow behavior when the host is following at a small distance the visitor. We also measure with Deep Squeak the ultrasonic vocalization, either during the social interaction, but also during the habituation phase when animals are facing for the first time the odor. And how did we get impact on social interaction? So animal was CD1, male and female, born in the lab and tested during the night phase. And to avoid using too much animal, we try to repeat the social interaction in changing the diet from one session to the other one. For example, with ambretolide, we have 5 male and 5 female. So we use, for example, this one was the host at the first session, and he began to be the visitor at the second session with another animal in the diet, of course. So how it looks like, we do habituation, 30 minutes, then social interaction. Then on the afternoon, we test odor preference from the host animal. And we have different groups for ambretolide, EBT, and vanillin. So as we know that session could be an issue, the repetition, and as of course it could have gender effect, we test that during habituations and during social interaction. So could odor value have an impact in behavior during habituation, and is there gender or repetition effect? So we don't see gender effect in habituation in the behavior, but repetition have a dramatic impact. If you look at rearing, ultrasonic vocalization number of USV or their principal frequency, what you could see is that there is an increased rearing from session 1 to 2, which means that, and you need to remember that the host here is new to the odor and new to the procedure, but here the hosts have already been visitor the first time. So they already knew the odor, they already knew the cage. So possibly they explore maybe more. It's not clear why. Maybe there is more exploration because less anxiety, or maybe they are less disturbed by the odor. We don't really know from now. But what was very interesting is that you decrease the number of ultrasonic vocalization clearly, which may signify that in a way this vocalization could have a meaning about the novelty of the odor if it's linked to the odor. So are the valence what we expect to be a value of the odor changing the behavior of animal? So if you look for rearing in different odors, so the negative one, the neutral one, and positive one, the only thing you see is that there is less rearing for ambretolide, but same kind of number of ultrasonic vocalization with a lot of variability, and also same kind of principal frequency. I don't show you the second session because it was, with the repetition effect, it was hard to have something to clear to look at. So this impact on rearing, maybe it's difficult to interpret because it could be also the diffusion of the odor, or I don't know. I'm open to suggestion because these results are very, very new. During social interaction, what are the impacts? So first, we have, as expected, because we have already done some social interaction with female, we have gender effect in the dominance part. So you see here, the male are less following than female in duration. They also do less vocalization, and it could be very consistent because we have demonstrated in this task that vocalization are really related to dominance. So the most you have domination or following, the most you get vocalization. And the frequency are almost the same. So it's interesting because here you get a gender effect that you don't get in vocalization emitted during habituation. About the repetition effect, we also have the important repetition effect. So, for example, in fact, the animals that are host on the second session are more dominant than the one of the first session. So you have more contact initiated, you have more number of follow, and less number of contacts initiated by the visitor. So it seems that in a way, having been visitor the first time give you advantage or make you more dominant during the second session. And it's not really changing the USV frequency or the number of USV, even if there is a lot of variability. But what about the other impact? So if you only look at the first session, as you could see here, there is not significant impact of the odor, neither in the contact initiated by host. On the following, there is something, but you see this point is really nothing, probably. Neither in the contact initiated by visitor. Ultrasonic vocalization are low, but still there, and frequency doesn't represent something, and odor preference is very variable also. So we think that, okay, we were expecting that probably odor may have different impact on different animals. So we decide to use principal frequency of ultrasonic vocalization because in mouse we demonstrate, and in rat also, maybe there is a link between frequency and affective value. So we decide to use this information of value of principal frequency as a way to suggest that if the value is, if the frequency is high, maybe the arousal and emotion of animal is positive. And what would be the impact on the behavior, and was this odor preferred or not in the prefrontal task? So what we decide to do is to reinterpret data in splitting animals in 3 groups regarding the frequency of USV during social interaction. And as you could see here, so we also consider both sessions, as the frequency is not changing that much during session. Of course, it's exploratory, there is a lot of possibility, but when you do that, so you split in 3 groups regarding the quartile, and like that, you have a group that utter low frequency USV, a medium group with medium frequency USV, and high frequency USV. And of course, we were suggesting, okay, if these guys have very high frequency USV during social interaction under this odor, probably this odor is induced positive affective state and gonna be preferred. And it's science, it's not exactly what we get. First of all, we were interested to know if there is a link between frequency during habituation and frequency during social interaction. As you could see here, no. If there is a high frequency during social interaction, there is not high frequency or no difference in frequency during habituation. Probably these USV are not conveying the same information. For dominance, it's interesting because as you could see here, it seems that maybe when you have a high frequency, you have low dominance. And if you only use animals who do following, if you do there is a negative correlation between vocalization and frequency of vocalization, sorry, and dominance, meaning that the low dominance, if you are very low dominance, you have higher frequency of USV. And eventually, most importantly, we get a significant difference in odor preference, which is great. But as you could see, it's not that clear, because for the highest frequency of USV uttered, you have almost not clear preference for this odor, and even sometimes avoidance. And for medium or very low frequency USV, you have very large either preference or avoidance. So it's a very complex impact we get. And if we try to, discuss that, I have think about this kind of discussion. So if you look at, so there is habituation, then social interaction, then preference of odor. So it seems that when animals have a high positive frequency and possibly positive affective state. There is low dominance, and they don't really get interested by odor, and they may also avoid sometimes the odor. And for me, it's because there is a very huge limitation of approach as, of course, as measure of some value of the odor. For example, I have found some papers showing that male in some situation could approach predator odor, male mice, in a task where there is some risky, male need to go through an odor, predator odor to get food. And in fact, the male are just waiting, are just stopping and looking at the odor after they get to food. So it's not that clear. For the negative or neutral vocalization, if it's real, there is high dominance and an approach of the odor. So it's not clear. Maybe there is some distraction. Maybe if the odor is maybe more salient for the animal, they are distracted in their interaction, and I don't know, there is a lot of things to dig into. But what is clear is that just with odor preference at the end and frequency of USV, we don't really get information about how this odor gets something, how the odor change emotion in animal. This work is in parallel with another work with Christine Bailey, where we also have tested maternal odor impact on social behavior, and we hope that with both this data, we're gonna have more idea of what is going on. And eventually, what was very interesting for me, and I was not expecting that, is that vocalization in different contexts, like habituation and social interaction, seems to be very different. Because we were wondering maybe odor during habituation could give us information about the affective value or the reactivity of animal to odor, but no. And also it's interesting because probably it's not conveying the same information because there is no gender effect here, but a huge gender effect with this odor vocalization. So it opened many questions. So eventually I want to thank the team of Sylvie Granon, our team from NeuroPSI, and especially Damien Legal and Pauline Pesset, who performed the experiment. Also the technician like Jonathan Pierre, who breeds the animal and keeps them in the lab. And of course, Christine Bailey team from Breed, who give us a lot of advice about how to use odor and what we could do with that. Thank you.